our speaker, Brad Shaffer, who's a professor at UC Los Angeles, UCLA, and also the director or principal investigator for the, let's see, what's the program? The California Conservation Genomics Project, which he's going to tell us about today. And he is, as he was telling us beforehand, he's primarily a herpetologist. I'm not sure if he's primarily interested in salamanders or snakes or lizards or whatever. Maybe he can tell us that as well. But he's interested in the effects of climate change on animals and other populations in the state of California. So that's what he's going to tell us about today. We're going to mute everybody while Brad talks and take questions at the end. But feel free during the talk to post to the chat that you have a question or whatever your question is, so that when Brad is finished, we can go to you directly, and if you want to unmute you and you can ask your question or a follow-up or whatever you want. So we don't want to interrupt him while he's presenting, but at the end he's open to staying as long as people have questions. So with that, Brad, you can share your screen and let her rip.
Okay, thanks. So couple of things for you quickly before I get started. First, McLaughlin is in an amazing place, and you should absolutely go up there. And Kathleen, you should make sure that late in the morning, your tour goes by some of the really nice ponds at McLaughlin, and you should bring your binoculars, and then you should look for basking Western pond turtles, because there's a really good population of them up there, and they are a candidate for federal listing right now under the U.S. Endangered Species Act. So you get to see a potential endangered species before it gets listed. And that right now they should be out and about and basking and stuff. So that's really good fun. Let's see. Do I have anything else? No. I think what we should probably do is get started, and I will share my screen. And I guess the one other thing I want to say is that if you have questions as I'm going through this is a lot of sort of genomics stuff. And Kathleen told me that this is an audience that's perfectly fine and perfectly capable of consuming genomics kinds of ideas, at least. So they are in here. But if something doesn't make sense to you, if you don't understand a term or something, I mean, just ask. And because I actually would rather be interrupted and straighten that out rather than lose people and therefore just be boring at this hour of the night. So with that, let's share the screen, and we'll share this baby, and then we will go and we'll see what happens. So what I'm going to talk to you about today, something Kathleen knows very well that I've been working on for the last year and a half or so, and it's this project which we call the CCGP, the California Conservation Genomics Project. So this is a slide, and some of this is material that I presented to the California Department of Fish and Wildlife, one of our, our big state regulatory agency for endangered and threatened species a little ways back. And one thing just to notice here is this little laundry list of people and where they're from. They are a collection, this is our executive committee for this project. So it's a collection of folks from different UCs. Those of you who know Berkeley might know Ian Wong. He's a just recently tenured professor at UC Berkeley. If you know UCOP, you might know Peggy Fiedler. Anyhow, it's a collection of folks who together with me have helped sort of develop and motivate this project. And so I just want to make sure that you sort of see that. Okay, so what's going to be our game plan with this talk? Here are the things that I think I'm going to try to hit on. So The talk is about the relationship between and developing and using genetics and really genomics data for conservation. So we'll start out by talking a little bit about why genetics or genomics is important for conservation. And that relationship has been crystal clear for decades. It's spanned most of my career. My career has spanned decades. So we'll talk a little bit about that. And then we'll talk more about why kind of business as usual, how we do genetics for conservation, in my mind at least, is not enough. It has sort of been very unsatisfying in many, many cases, not all, but a lot, and how we can do better. And how the CCGP, the California Conservation Genomics Project, many of us feel can change the game. And it can do that for both basic science, the sort of science of landscape genetics and landscape ecology. And it can also do that for applied conservation, for how the California Department of Fish and Wildlife or the Wildlife Conservation Board makes decisions, or the Nature Conservancy makes decisions about conservation. And then I'll talk to you some about where we're at in this project. We're a bit over a year into it, and just give you some results, or at least some progress. It's not really results. So one slide on why genetics is important for conservation. And again, for those of you who know stuff about, who are kind of conversant in genetics, hopefully this will make sense, and I'll try to explain it for everybody. So genetic variation comes in two flavors, right? There's what people refer to as neutral variation. And that means, remember, your DNA is a code, right? A's, C's, G's, and T's strung out. Humans have 3.2 billion of them that code for you. And turtles, those turtles up at McLaughlin have 2.6 billion. A fruit fly has about 100 million. So different genomes are different sizes. Most of that sequence information and most of the variation that we see in it, so a difference between Kathleen and myself in our sequences for a particular gene, most of it is largely viewed as neutral. And so what does neutral mean? It just means that if you got an A at that position or a T in that position, it doesn't make any difference, okay, in terms of your fitness or what you look like or anything else. So that's neutral variation. And then obviously there is some variation which kind of codes for, for meaningful variation, adaptive variation. And that's the kind of genes that matter, if you will, or mutations that matter. So neutral variation, there's way more of it than there is functional variation or genes that matter. And neutral variation is what we use in population genetics to infer just about everything. Okay, and the reason for that is that neutral variation, because it doesn't matter in terms of fitness, neutral variation sort of evolves in a pretty regular way. So you can base a molecular clock on neutral variation. You can measure meaningful levels of migration between populations with neutral variation. Whereas genes that matter, natural selection kind of screws up all of that, and it changes rates of evolution, it changes fixation rates, it changes all kinds of stuff. So neutral variation is actually the sort of core of an awful lot of the ecological inferences that we make from population genetic data. And so among other things, we use neutral variation in genetic data sets to infer population structure. Or is this one big panmictic population where everyone mates with everyone else, or does it composed of a couple of subpopulations where there's random mating within but not between? Gene flow is very tightly associated with that, right? Our populations north and south of San Francisco Bay exchanging migrants, exchanging genes or not. And we can get at that with neutral variation. Hybridization, if we have two closely related species, are they hybridizing or not? We can look at the movement of those genes between them. Demography, this is a big one. So NCB is a term that you may or may not have heard of. It's called effective population size. And this is the parameter that, so capital N always sort of refers to a population size. And effective is it just means that it's more or less the number of individuals who are breeding and producing offspring in a population. So if you have a population of 100 individuals and 2 of them mate and make kids and the other 98 don't, the census size might be 100, but the effective size might be 2. Okay, and that matters a lot because the effective size is very closely linked to the amount of genetic variation that's in the population. If only a couple of individuals are breeding, there's less genetic variation. If everybody's breeding, there's more. And then standing genetic variation. So that just means, is there a lot of variation in a population or is there hardly any, and is it really inbred? And hopefully everybody knows that when you have inbreeding, that can lead to inbreeding depression, that can lead to populations of unhealthy individuals. And critically, with coming back to the idea of like climate change and resilience to climate change, there's a sort of very strong and I think generally well accepted and well supported argument that populations that have a lot of genetic variation have a lot of capacity to respond to change. And that's just true. It almost has to be by definition. And so what it means is that if you have pockets and areas where there's lots of variation within populations and other pockets where there's hardly any, those former pockets are going to be much more likely on average to be able to respond to climate change or respond to pesticides or respond to a lot of things that we do to populations, whereas those with very little will not. So those are some of the things that neutral variation allows us to get at. And then genes that matter, so where there's been a lot of localized selection, natural selection going on, those are genes that really help us understand local adaptation. So oftentimes those are genes that have been selected out during the process of sort of resilient populations responding to something, right? Those are ones that show relationships with climate when you go up a transect. Those are ones that you know, there's heat tolerant and cold tolerant genes and stuff like that, or alleles of a gene. And they also matter, but they've received much less sort of work than neutral variation. So anyhow, that's just sort of some of the context for why genetics is important for conservation. You know, we oftentimes want to understand things like population structure, effective population sizes, standing genetic variation. And then as a conservation biologist, we want to protect that variation and we want to maintain it. If there's natural variation north and south of the Bay Area, we want to maintain that and not screw it up by moving things around and that kind of thing. Okay, so traditionally, the ways that we use landscape genetics, and by landscape genetics, I mean that it's studying genetic variation, usually in a very small part of the genome of an organism, across a landscape. So lots of individuals across a landscape. And traditionally, we use it in the context of conservation to quantify barriers to gene flow. So again, you know, are the Sierras a barrier to things that occur west and east of them? Are the Tehachapi Mountains, which are a major barrier to lots of species, which sort of separate the southern San Joaquin Valley from the Mojave Desert? Is that a barrier to gene flow and differentiation in a species? And then to kind of use that information to delimit so-called conservation units. So we might take a species and say, well, there's 6 different units in it and that are genetically very differentiated from each other. And we want to maintain the integrity of those units. We sort of want to protect all the variation in the species. And that means we're going to protect populations within each of those units. Okay. And it makes a big difference if a species has 1 unit or 10 units. If it has 10 units, there's a lot more you need to protect than if it only has 1. If it only has 1, just as long as it occurs in some protected landscape, you're fine by that line of argument. That's not the only line of argument, but it's a very important one. Okay, so I said I wanted to talk about business as usual. And that just means kind of telling you how we have traditionally done this kind of work, and sort of the kinds of inferences people have made. And I have two different kinds of examples for business as usual, using genetic data to think about landscape genetics. So business as usual, number one, this is a paper that was published a few years ago by a couple of colleagues of mine. I was not a part of this study, in the Santa Monica Mountains. The Santa Monicas are the largest urban national park protected area in the United States. And I don't know if they're the largest urban one in the world. 160,000 acres of open space, surrounded by, in large part, by either LA or the Pacific Ocean. So there's a map of the Santa Monica Mountains, okay? And here's a more realistic map of the Santa Monica Mountains, okay? So the whole thing, everything that's outlined in this dark green, is part of the Santa Monica Mountains National Recreation Area. But it has in-holdings of all kinds. The National Park Service owns some of it, state parks own some of it, county parks, you know, everybody and their brother owns some bit of this thing. And the National Park Service tries to kind of manage it. Well, now, one of the things that is sort of a big feature of the Santa Monica Mountains, and if we were in person, I would ask everybody to show their hands to tell me if they've been there or not, is that the 101 freeway, which those of you in the Bay Area also know, it kind of runs right down the middle of the Santa Monicas. Okay, it runs along sort of through the San Fernando Valley and out towards Thousand Oaks and Ventura County. And the question that was addressed in this study is the 101 freeway a barrier to gene flow and movement of organisms in the Santa Monica Mountains? So what did they do? Well, so here's the first study. They went through several different species in this paper. And one is a Sidewatch lizard, Usda stansburyana, for those of you who care about such things. A very small, active little lizard. You can see here, there's the side blotch under its armpit. And there's the 101 freeway, which they wanted to understand. And here's a bunch of sampling sites. And the blue dots are where they had a one or a few side blotch lizards. And they used a technique, it's called Microsatellites. So they got 20 or 30 genes, 15 or 20 genes probably. No, in this case, I think it was about 10 different genes. And they used those genes to try to ask the question, is the 101 freeway a barrier to movement in side blotch lizards? I mean, it presumably is. It's hard for them to cross the freeway, but still, can we see a signal of that? Right, that didn't work. Okay, and here's kind of what it looks like. This is using one of these landscape genetic interpolation maps to look at sort of two different regions and sort of where they kind of line up, where the boundary is between those two regions. So there's kind of this green region over here and this red region over here. And, you know, if you can sort of remember, that's where the 101 freeway is. It's still in this next map. And, you know, it kind of looks like it lines up pretty well, right? So, yeah, maybe the 101 is kind of a barrier. But the sort of the epicenter of that barrier is actually down here. It's this white thing. And that's actually kind of far south of the 101. And the other thing you might notice is that the sampling north of the 101 is pretty good. There's a bunch of blue dots. The sampling south of the 101 is really lousy. There's only 3. And there's tons of lizards there, but they only have 3 sites. And it's far away from the 101. So you know, do I believe it? It's a small number of genes. You know, it's probably 0.001% of the genome. Maybe they're a little bit funny genes. The sampling's a little weird. Maybe I believe it, maybe I don't. Maybe there's something else going on. They put the 101 in the middle of the San Fernando Valley. Maybe it's just the San Fernando Valley, and this is more where the valley is. So you kind of make an inference, but it's a little unsatisfying. Okay, so then they repeated it in the same study with a bird called wren tit. Wren tit is a chaparral specialist, They hate open spaces. They don't like to fly across lawns. They don't like to fly across anything. They're very, very sedentary, non-migratory little birds, and they really hunker down in chaparral. Here's the sampling, and you can imagine if I didn't like the Sidewatch sampling, how I'm going to feel about this, because these samples down here are way down here. But here's the 101 freeway again. And here's what we get with the wren tits. And again, we sort of see a difference between the north and the south, but there's an awfully big sampling gap, and it doesn't quite line up with the 101. It's only a few genes, and so it's kind of close, but it's sort of unsatisfying. Okay, well, here's a third one. These are bobcats, okay, and bobcats are widespread throughout the Santa Monica Mountains. There are a lot of samples. My colleague Seth Riley was the person behind this bobcat sampling. And when we look at it, you see over here, I mean, here's the 101 freeway, and you go, bang, that's what I'm talking about. Okay, there's differentiation that comes right up to the 101 freeway, and then the other color is sort of on the other side of the 101 freeway. Except all of this, this is all kind of inferred to be the north of the 101 freeway, but it's south, okay? And, and well, what about over here? Well, they couldn't get any samples over there. So, and again, it's a small number of genes. It sort of fits, but doesn't fit. So, what are the points? It's not very satisfying in many ways, and they're all different, okay? And they all use different genes, and they all use different sampling strategies, and they all use different everything. And so when you sort of want to tell a story, in many ways, I think you have too many degrees of freedom. You can sort of talk your way out of the anomalies. Things aren't consistent. You maybe don't have quite enough data. If you had more data, maybe these would, this blue wouldn't come down here. It's sort of unsatisfying. Okay, so that's kind of one form of business as usual in landscape genetics. Here's another, okay? And by the way, that stuff on the 101 freeway is a lot of the data that's being used to motivate putting a wildlife overpass over the 101 freeway, which is going to cost $100 million. And those are the kinds of data that are supporting it, okay? I mean, they're fine, but maybe they could be better for something as big as that project. So here's another one. This is at a very different spatial scale. This is sort of at the spatial scale of California. So this is one of my favorite organisms on Earth. It's called a Spadefoot Toad, but it's not a toad, so we just call it a spadefoot. It's completely unrelated to toads, but it's a kind of a neuron. It's kind of frog. Wonderful animals. This is a full-size adult. They're nocturnal, so they have vertical pupils like many nocturnal vertebrates do. They're tremendous animals. They're just wonderful. Spadefoots are currently up for listing at the federal level under the U.S. Endangered Species Act. And a graduate student in my lab who finished up last year, Kevin Neal, did a lot of work on spadefoots to sort of try and collect much better genomic data, in this case using 20,000 genes instead of 20 genes in the previous studies to study spadefoots. And so, what did Kevin do? He and I went out and, well, plus also I've been doing this for years. We collected spadefoots. This is their range. They're completely restricted to California and northern Baja. And we collected them from, every inch of their range where they still exist. They're in a lot of trouble in some areas, but everywhere where we could get them. And then did the same kind of landscape genetic analysis to look at patterns. And what I'm showing here is just a section. This happened to be a figure that I had that I could pull for this talk from the area from the San Francisco Bay Area here. There's Monterey. Here's Point Conception, Santa Barbara County. Okay. And for that part of the northern group of spadefoots, what Kevin found is that there was a break at San Francisco Bay, basically, that goes across the valley. Okay, one group there and one group here throughout the rest of the valley. And then this area of northern Santa Barbara County was a sort of unique entity. Okay. And that's interesting, and that's being used right now in discussions about managing the Spadefoot, and should we have the happy conclusion in my mind that it gets listed, these will become management units and recovery units, I think, for the Spadefoot. Well, let's compare that to another species, which we've also done in our lab. This is work by Aaron Toffelmeyer, a student who finished it about a year ago. And this is on an amazing lizard that many of you may have seen called an alligator lizard. This is a southern alligator lizard. And this is a pair of southern alligator lizards. So there's one, and there's the other. Her head is down there, and she's kind of wrapped around here. Whoopsie. And this is a picture that my wife Louise took in our backyard. We live in very, very urban LA, and alligator lizards, among their many other wonderful attributes, are the most urban adapted native lizard in California. So you can get these things anywhere in LA. And one of the things we're not right now, but one of the things we're trying to figure out is how in the world they survive. In LA, they're in the Bay Area. You get them in Berkeley, you get them wherever. They're really pretty amazing animals. Anyhow, that's a picture of a pair of them, and that's their courtship. Right? He bites her head. That's how they court. Anyhow, so when Aaron did this work at the range-wide level, again using roughly sort of 8 to 20,000 genes, depending on the analysis, what she found is that there were 5 genetic units of alligator lizards, and that she did this study. We're in the process of publishing it, and there's these different colors correspond to the different units in the species range. So if you compare those two, and what I've done is clipped the alligator lizard map down to the same as the Spadefoot map, what do you see? Well, again, you see that the Bay Area seems to be a really major discontinuity between populations to the north and the south. And you see that in the alligator lizard, and you see that in the Spadefoot. Alligator lizards seem to show another break around Monterey Bay. So kind of, Santa Cruz County up to the Bay Area, up to Solano County, and then south in Monterey and San Luis Obispo County, and maybe over in San Bernardino County here. Okay. Spadefoots don't show this break. And they show another one kind of down here in Santa Barbara County, this isolated patch in Santa Barbara County. Well, the alligator lizards don't, but there's another thing south of that in Southern California. So maybe that boundary is a little bit off, and maybe that should be pushed up here a little bit more, and then they would kind of match up. So they kind of match up, they kind of don't. Our sampling is much denser for alligator lizards than it is for spadefoots. We use different genes. Again, it sort of kind of works, but it kind of doesn't. They're separate studies. They were done with separate goals in mind, et cetera. So what are the problems with this approach? Well, in my mind, at least, the problems with this kind of piecemeal approach, we'll use microsatellites on this, we're using RAD-seq on that, we're using targeted capture on another, we're throwing in a little bit of genomics, a little bit of this and that. The problems with this approach have to do with the fact that the data between studies, they're not combinable, they're not compatible, and they're not consistent. They're just, everything is different. And so when you see differences, you don't know what to attribute it to. In all of these cases, it's just trust me, with these kinds of data sets, it's almost impossible to add new data to them. And that's because they're using a very small part of the genome, and it's very hard in the next study to get exactly that same small part of the genome. And so the study is done, and it tends to just sit there. People don't tend to add to it. And it lacks information from the entire genome. So it lacks information on neutral versus sort of adaptive genetic variation. Again, adding to this kind of lack of combinability. And what you often end up coming away from these kinds of studies when you're sort of trying to compare things, but they weren't done in the same way, is wondering whether differences and similarities are idiosyncratic, sort of just due to luck, or are they general? Okay. So what's our approach? So that's all kind of background. And this has been my life for 35 years as a University of California professor of one sort or another. And to my mind, the solution is don't do it piecemeal and don't do it with scraps of the genome, but do it right. Use, and what does that mean? That means, and this is my cool animation slide, do it on lots of species. Do it on acorn woodpeckers, do it on pond turtles, do it on pipevine swallowtails, do it on freshwater crustaceans and great white sharks and black rails and song sparrows and sea stars and oak trees and rattlesnakes and black bears. Do it on everything, or as much as you can. Okay. And what's more than that, don't just study lots and lots of species all at once, but use exactly the same techniques and use the best techniques that money can buy. So study the same part of the genome. In our case, that part is the whole genome. Okay. Study with sort of as best as you can similar sampling across the range of each of these species and analyze everything in exactly the same way. And then a lot of this idiosyncratic stuff that you worry about, in theory, goes away. So that's the CCGP. So at a glance, the California Conservation Genomics Project, where we're at right now, is we have about 245 species that are organized into 135 or so projects. So a project just means it might be one species or a couple or three closely related species that we sort of count as one. The strategy is for each one of those projects, we put together what's called a reference genome. So a reference genome means you take one individual and you build the best, most beautifully assembled and compiled genome that, within reason, money can buy. So something that's on par with it, with an assembled human genome. And you do one of those for each species, and that serves as a template. That serves as a tool that you can use. The guts of the project are, for each one of those species projects, we sample 150 individuals, more or less. That gives a pretty good spread across the state of California for these species that are as widely spread out as possible. We choose and curate the species carefully so that they cover a lot of the ecoregion and ecological variation that's found in the state. So we sequence 150 individuals spread out across the state per project. And we sequence those at the whole genome level. So we don't do 5 genes or 50 genes or 500,000 markers. We do, if it's got a 3.2 billion base pair genome, we sequence that thing, 150 of those. If it's got a smaller one, we do that. So it's full genome data. So that would, if you just do the arithmetic, that comes out to something a little south of 19,000 genomes that we assemble for this project across roughly 135 species. Some of the features of the project are that we've set up in the last year. We've set up a dedicated core facility at UCLA so that if Kathleen is an expert in, I don't know, whatever she's an expert in, oak trees, but she's not really great at genomics, we'll do it for you. Okay, you just send us the samples sampled from across the state, and we'll set it all up and do it for you. We get uniform genomic coverage. For those of you who understand genomics, the way you have confidence in your sequencing of a genome is you just sequence it over and over and over again, and that's called X coverage. Okay, if you sequence on average the whole genome 10 times. So for a human with 3.2 billion base pairs, if you made from me 32 billion base pairs of data from my genome, randomly assemble, sort of randomly sequenced across my genome, that would be 10x coverage. You know, if you did 320 billion reads, it would be 100x coverage. So we're doing uniform full genome coverage at 10x. So each sequencing each of those 150 individuals for each of those 135 species 10 times. And then we're hiring, we're in the process of doing this right now, some really good bioinformaticians and landscape genomics postdocs who will make common analysis pipelines. So when those data come off the sequencer, they go right into, you know, this postdoc's pipeline that gets analyzed. So they're all analyzed in exactly the same way. The, you know, filters that you use, the decisions you make on whether you're calling a gene or not, or calling a variant or not, they're all the same for everything. So the data are all compatible and combinable. Okay. Now the data also go to the principal investigators who are involved in these projects, and they can do whatever they want with them. But for the CCGP, we're doing a sort of parallel set of analyses that are all the same. So all those issues about data combinability and compatibility and comparability are in theory sorted out and solved. And what do we hope to accomplish with doing this project? Well, we want taxonomic and ecological coverage for the state replicated across many species. So what we want to be able to do is for each species basically develop a kind of heat map of genomic variation across the state, where we can see hot spots and cold spots, places where there's a lot of variation, places where there's little. What are the barriers to gene flow? What are the different units, genetic units across the species? How important are the hatch bees compared to, you know, whatever else the coast range in structuring genetic variation? So develop meaningful combinable analyses of variation across the state. And the goal in many ways is to say, well, so for example, if we buy into the notion that hotspots of genomic variation are on average more climate resilient, you know, more resilient to falling apart in the face of rapid climate change, are there certain regions that for species after species after species, it doesn't matter if you're a freshwater fish or an oak tree or a spadefoot, those are just regions of the state that have maintained a lot of genetic variation. And if so, then those are, in our minds at least, hotspots. They become critical lands to acquire and protect. So we can go to the Wildlife Conservation Board, we can go to the Nature Conservancy, we can go to the people who deal in these kinds of landscapes and say, you know what, you know, we need this because this is what's going to keep stuff going in 50 years when the climate has changed radically. If there are several such hotspots, we also want to have corridors that connect them. We want to give organisms the ability, as they evolve, as they sort of come up with the genes that matter, the genes that are important for adapting to that climate change. We want those genes to be able to move. We want them to be able to get out into other places, right? And so that means you need corridors. And that's the goal of it, okay, in a nutshell. I mean, there's lots of other goals, but to my mind, that's the really meaningful one. So how do we accomplish this? How are we proposing to accomplish this? How did I pitch this to the state of California? Well, the way it's played out now is we have a team of about 100 principal investigators. Not every principal investigator, but every project is led by a University of California faculty member, or faculty, or a person who has principal investigator status at UC. All 10 UC campuses, including UCSF, is involved, which I find astonishing. We have that executive committee that I showed you on the first slide, which includes individuals from 5 different UCs, from the California Department of Fish and Wildlife, the US Fish and Wildlife Service, Governor Newsom's office. And so that's our executive committee, and pretty much every decision that we make, I mean, I may pitch them, but the executive committee approves it or doesn't approve it. We've met now 25 times in a year and some change. So we meet, basically every couple of weeks. And we have just ground through the whole process of how we're going to do this together. We have several hundred co-PIs. And then the California Department of Fish and Agriculture is a partner that we're going to be bringing in, that is still in its infancy, to bring some pest and agricultural species into this mix. The funding is interesting. Basically, I, with some input from a couple of other people, made a pitch to a member of the state legislature, the state assembly, and for the 2019 budget, we made a pitch for this project. We said this was really important. This should be a big system-wide initiative, and that member of the state assembly was impressed, liked the idea. The bottom line, after it was all said and done, is that we were written in as a line item in the budget for $12 million. And about 10 of that goes directly out to those 100 PIs. So we developed a whole process, which I could explain to you if you want, about how that all works and how we evaluated proposals. We had an external evaluation committee that evaluated every species proposal that came in. And the whole project is a really interesting mix of, on the one hand, being super tightly focused. So, we said we want 150 individuals from across the range of the species, want a species to have some conservation impact. We want, But then you, in 300 words, pitch to us why we should have that species and not another one, and why you're the right lab to do it and not another lab. And so it's sort of a combination of people pitching their taxa and us making, so a lot of individuality in terms of the taxa, but also a very sort of strict, rigid framework for how the data would come in. And it's worked really well. Some other features of it that are just interesting for those of you who follow this sort of thing is that there's no indirects. So that 12 million is all spendable. That's all green and folding. And that's from a PI's point of view. And that's because it's state funding, and the state said, well, we already fund UC. So we're not going to give any overhead on top of that. So there's no indirects. And there's no faculty salary because the state already pays our salaries. And so what it means is that you've got campuses and departments who are supporting this, even though they're not getting any support, they're not getting any indirect support. We're getting VCRs, vice chancellors of research, who accept this money with zero indirects, which is rare. And we have faculty who are lining up to do it, who aren't going to get any salary out of it. And that's kind of interesting. I think it's good. I think it's great. Okay, the data. Just some details on this. So the reference genomes, those super high quality genomes, we decided those were such a critical component of this work that we didn't want the PIs messing with it. So what we said is that you have to send us the world's highest quality tissue sample, because it's really important with these reference genomes to get high quality tissue. So you send us super high quality tissue and we will do everything else. It will not come out of your budget. It will not be, anything that you applied for. You just send us that oak leaf or that garter snake or whatever it is, and we'll do all the rest. And that's all being done, a combination of core lab at UC Davis, core lab at UC Berkeley, a research group at UC Santa Cruz, and a group down here at UCLA to sort of put all that together. And I can explain it to you if you want. So those are being done, by us, no fuss, no muss. All you got to do is have the tissue. The resequencing is being done to maximize landscape coverage across the state. And most, but not all projects have some conservation value for sensitive species. So a lot of times it's things like it's some plant that has that is widespread, so it gives us broad coverage across the state, but it also has an endangered subspecies. So it's going to tell us something about the genetic health of that little endangered subspecies, as well as the broad coverage across the state. It's that kind of thing. And then there's some which really don't have much in the way of conservation importance, except the fact that they're just super important ecologically. So they're oak trees, which they're not going to go extinct. You know, valley oak's not going to go extinct, but it's a foundational ecological species that we just want to make sure we really understand well. Okay, so a little bit more here. So you know, how are we doing on this? So here's our base map of California. And these outlines here are the 19 USDA recognized ecoregions of the state. And one of our goals has been to have sort of broad coverage across those ecoregions. You can also sort of split the coastal waters into 3 different ecoregions. So this is a little out of date. We have a few more than this now, but here's our coverage. So those are the individuals across species all pulled together for our marine. You know, we've got really good coverage of marine ecosystems. There's our plants. Okay, so that's trees, you know, herbaceous, shrubs, whatever. There's our plant coverage. There's our vertebrates, and those are tucked underneath the plants. So in terms of those dots, or at least some of them are. So anyhow, the vertebrate coverage is really good. As you might expect, there's a lot of good vertebrate folks, and invertebrates, which is insects as well as, you know, terrestrial mollusks and other things of conservation concern, are also pretty good, and they've improved the most from this map. So they're actually somewhat better than that. So if you take that map, to my eye at least, it looks pretty darn good. If we can get all of those genomes all sequenced, all mapped, all done in the same way, I think we're going to have amazing coverage. There's, you know, the one area that we're a little light on is the Mojave Desert over here. This sort of triangle is the Mojave, and out here in southeastern California, like out in Imperial County. But part of that is just that the diversity is lower out there. And species that are out in these areas tend to be very specialized, and we want ones that have a broad range. But overall, it's pretty good. I mean, the other is the San Joaquin Valley, but you know, there's nothing left in the San Joaquin, so you know, we did the best we can. In terms of numbers of species in each of those ecoregions that at least we have at least some sampling in those ecoregions, you can see it's really good. You know, so even in our underrepresented ones like the Mojave, we've got 56 species that are represented out there. You know, as I said, the Colorado Desert in southeastern California and sort of up here in the Modoc Plateau and stuff is a little on the light side, but you know, they're small areas and they're not very biodiverse. But overall, I think our sampling is really strong. Where are we at with this project? A year and some change into it. Well, the funds have almost all been distributed. In fact, today I have on my list we just had our final kind of third call to fill in some sampling and phylogenetic gaps in our sampling. And we have 5 more projects and I think about 10 more species that we're going to be funding. So going to be pushing those out tomorrow, and then I think the funds will be distributed. The species and localities have all been determined. In terms of those reference genomes, out of about 130, we have 87 of them that are in our pipeline. The first one is finished this week, but they'll go fast once they get going. So we have something like 2/3 of the reference genomes in play right now, about 1/3 of them still to go. My goal is to have 100 of the 130 reference genomes and 7,000 of the resequenced individuals done by September 1st, and the data completed by the 1st of July in 2022. The project is on a short timeline. We want to get the whole thing done in 3 to 4 years to be able to deliver that to the state. And I think we can do that. I'm a optimistic person. Show you a little bit more about sampling. So on the left is the sampling that's been done. That's, and again, these are a tiny bit out of date, but that's about 8,300 samples that are in hand, about 4,000 that are still to be collected. You can see both of them cover sort of the range of the state. Filling this in has been problematic for two reasons this year, because this was the year to fill in all of that and get all those samples. COVID hasn't helped any, as you might imagine. You know, we barely still have permission at UCLA to even go out in the field, even if we go by ourselves. And the fact that this is one of the all-time record droughts isn't helping any either, because you know, you need, for plants, you need fresh new growth of tissues, and you know, a lot of plants are just shutting down for the year because of the drought. But we'll work on it, and that's just sort of puts them both together and shows them in the ecoregions. This is another thing that we've done that's part of this being sort of a network project, a community project, if you will. So there's kind of sampling of all the different species projects, and Ryan Harrigan, who's a very accomplished GIS guy who we have as part of our team down here at UCLA, he put together this great algorithm where he said, okay, you know, if that dot right there is Brad wanting to get a turtle from that site, what are 5 other folks who have other dots that they need to sample within a 5-kilometer radius of that dot, who might be going out to collect something? And maybe Brad and those people could work together on it. So he put together this map, and each one of those dark circles shows the sort of radius of area where you have to go out in order to pick up several other dots. And I think the number was 5. And then we sent out to all of our PIs a reference database of all of this, so that if I'm thinking, oh, I got to go up to Shasta County, but maybe someone else is going up there, it tells me who to get in touch with. So it's a pretty cool resource that I'm hoping people will use this year. So that's just explaining that. How we hope the data will be used, we hope. So in my mind, the most kind of important and interesting aspect of it, that to my mind will be the sort of biggest home run, is to construct a statewide guide to sort of hotspots of genomic variation, genomic resilience. And if there are consistent hotspots that go across lots of taxa, I think identifying those will be a huge win in terms of then identifying lands that should either be acquired or managed differently than they are now, or a justification for managing them as incredibly important protected areas. We can use it to enable forensics and repatriation. We can use it to think about, as we have done with a different study with desert tortoises, about things like, where do you put giant solar panel farms so that you get the green energy that you want, but you don't screw up the environment. You don't plop it down on top of a really important population or region of the state, and providing a lasting resource. So just real quick, because we're going on a bit here, I'll just run through this. So in terms of variation, again, this is a, you know, this is a not a CCGP study, but another one that we did on a species called the Foothill yellow-legged frog that was recently listed by the state of California based on this. And you know, there's just a sort of map of hot spots and cold spots. It's in this case, these are areas of higher genetic variation. These are areas of lower genetic variation with the warmer colors. And you know, can we develop these not just for the 10,000 loci, 10,000 genes that we used with Foothill yellow-legged frogs, but can we do it for the entire genome for all of those species and stack them all up so that we can see what kind of patterns we see across the state. Maybe there won't be any. Maybe everything is idiosyncratic, but I bet not. I bet there will be regions that are real hotspots for variation and really need protection. And interestingly enough, I bet they won't be in the places we protect. Because what do we do, as a culture and as a state? We protect the Sierras. Well, the Sierras, were under glaciers 20,000 years ago. Everything that's moved in there is all really, really recent. And recent migrations don't tend to harbor a lot of genetic variation. Where are the places that have been stable and have really, really long-term stable populations? They are often in the sort of stable lowlands, places like the Great Central Valley. So I think it's going to be really interesting to see how that plays out and how those maps, those patterns of genomic variation map to the places that we protect. Because we protect pretty places. I don't think we protect biologically the most important places. Forensics, this is just an example that, that many of my colleagues are really interested in. This is a Western pond turtle, a species that I've studied a ton, and that again is up for federal listing under the Endangered Species Act. And one of the things that happens with turtles and with succulents and with lots of plants and also with marine life is that there's poaching. There's illegal movement of animals and plants, and people don't know where they came from. Or there are these funny, weird populations of them that are established somewhere, and people want to know, is it native or not? Or is it an introduced population? And if it is introduced, where did it come from and how did it get there? And use that in trying to manage it. But the main, and that's the example that I happen to have. So the example real quickly is Western pond turtles at Edwards Air Force Base. So for those of you who don't know, this triangle defines the Mojave Desert. There's the Tehachapi's there, there's Bakersfield. And Edwards is a huge Air Force base stuck out in the middle of the Mojave. And this is not good freshwater turtle habitat, okay? And there's a population of pond turtles that showed up there in a wetland. And the folks at Edwards want to know, is this native? Is it non-native? Should we be protecting it? What the hell's going on with this stupid turtle? Well, there's our kind of sampling for Western pond turtles. And we're in the process of doing this as a CCGP species right now. And there's kind of our coverage for 150 turtles. And there's Edwards again. And the things that we can resolve very quickly with this, with the Edwards Air Force Base turtles, are those related to, and did someone just sort of bring some turtles when they were driving down the 99 and came over the hill or down the 395? Did they get a turtle from over here in the Sierras and dump a few of them out at Edwards? Is that the source? Did they come from the coast? That's also pretty close, and somebody, easily could have on their way out to Vegas, dumped some turtles out in those wetlands from the coastal ones. And these are, we can easily differentiate that. Or interestingly enough, in the Mojave River, there are some native vestiges of pond turtles. There's a couple of populations of them, a place called Afton Canyon and one other spot that has Western pond turtles that are native. And maybe those Edwards Air Force Base turtles really are the native ones. If they are, they're one of the most endangered populations of pond turtles we have in a species that's a candidate for listing. If they're not, they're an introduced exotic. So we can understand that. And by the same reasoning, if these were not turtles at Edwards Air Force Base, but if these instead were poached turtles that were picked up at LAX, and there are hundreds and hundreds of shipments of thousands of turtles that go out of LAX every year, we could tell them, where are the poachers getting their turtles, right? We could sequence them up and tell them where they're coming from. Is it, coming from up here? Is it coming from down here? Where are they coming from? And you can do the same thing with abalone. You can do the same thing with, with sea urchins. You can do it with, whatever is being poached. If it's a CCGP species where we have that base map. So that's kind of a cool thing. And then I think maybe the last thing or second to last thing I'm going to talk about just for a minute, and then I'll wrap it up, is where do you put a solar farm? Now we've done this exercise with Mojave Desert tortoises. This is one I just pulled this map out from a paper that a colleague of mine, Victoria Sork, did on again, genetic variation, not at the whole genome level. This is using some repeats. It's a different kind of data set on Valley Oak. And if you just look at this, these pie diagrams show you sort of how much variation there is within a locality, okay? And you can see there's actually a fair bit of variation right here. There's sort of red and blue, there's red right next to blue in this area. And that goes from around Point Conception over into Western Kern County. And Western Kern County is a total hotspot for dumping these giant solar farms. And why is it a place to do it? Land's cheap. Everybody thinks of it as just sort of part of the San Joaquin Desert. It's not very interesting. It's politically a climate that's fine with paving over the land if they're going to get some money. There's a sort of, you know, that happens there. So that's an area that it just so happens has a very high level of genetic diversity for Valley Oak in that region, you know, based on this map. And it's an area that's sort of prime real estate for chopping down the trees and paving it over. And you know, the question is, if we had that kind of data really well resolved for Valley Oak or Coast Live Oak or whatever it was, might we be able to use this to convince the regulatory agencies, even in a place like Kern County, that, you know, maybe they should nudge it a little somewhere else, you know, sort of pop it over here into, you know, into Eastern Kern County, if I'm not too far north, or, you know, a place where there's less variation and the population's a little less important. Is that something that we could do? And I think we could. And this is just to say that the last sort of pitch for this data set is that it really is a lasting resource. And it's why we're doing everything at the whole genome level. And the reasoning there is that there's this annoying tendency with genetics research that just when you finished a project, the next genetic tool comes along and it gives you access to more of the genome and more information, and say, oh, well, I better redo it again. Well, as my colleague Chuck Langley up at Davis said to me years ago, he said, you know, the beauty of whole genomes is that there isn't any more. That's all there is. And so it becomes a much more lasting resource. And there's a lot of good things about that I can talk to you about if you're interested in. But it becomes something that feels like it ought to be a more long-lasting resource because it's the whole genome. It's all there is, and there ain't no more. There's some problems to resolve. How do we make that resource available? You know, these are enormous data sets, enormous. And you want to make it not just that it's out there, that people can, like myself, can analyze it and use it, but you want to make it quickly, easily, flexibly available. And that's going to be part of, you know, in the next couple of years, working with computer scientists to try and figure out how to do that. Make it easy to use, easy to query, and easy to interpret. And by doing that, my goal is that we can convince decision makers that this is an important resource for them to use. And you know, my example that I always say to people is that if we had that resource, so two properties that you might have heard of that are very, very important properties down here in Southern California. One is called the Dangermont Reserve. So that's the big 36,000 acre ranch that the Nature Conservancy just picked up that includes all of Point Conception in Santa Barbara County. And Dangermont is a big, huge, interesting, important acquisition that TNC made. It cost them $160 million and they bought it. Well, there's another property due east of that called Tejon Ranch. and it's way bigger. It's 250,000 acres, and Tahone could be on the chopping block, and it could be on the chopping block for that kind of money. And if TNC was trying to make a choice between Dangermont and Tahone, I mean, how do you decide? Well, this might be one way to decide. Is Dangermont a more of a kind of hotspot across lots and lots of species, or is Tahone? I'm not saying they would use that to make a decision if that were available and if that were possible, but that's my hope. And I want to be able to make it, the resource available so that a solid TNC administrator could go onto our website and in 5 minutes get a sense of whether Dangermont or Tahone harbor more variation species by species or across, the 100 species that are on each of them. I think that would be a real resource that would be really useful. We have built a website, and this version 2 of it, we just launched a couple of weeks ago. If you just Google CCGP, it pops up first now. I'm very proud of that. And it's a website that we had a first version. This one is a much prettier version that has sort of resources for scientists, for policymakers, and for what we call curious conservationists. That is the public. And we will be fleshing that out and increasing it, but you can see the species that we're covering and some of the resources that we're developing on that website. That's as much as I have to say, and I certainly prattled on for a while. So I think I should call it there. And if you have any questions, I'll stop sharing my screen. I don't know if you do or not, but if you
yeah, we do have some questions.
Okay.
Maybe I'll start with someone who posted a note early. Lisa Krieger, maybe you can unmute.
Yeah, I'm silly question. This is elegant science, but how do you get your genetic samples? Do you have to kill the animals to get them?
No, I study reptiles and amphibians. So the honest answer, Lisa, is that for the reference genome to do it well, you have to kill the animal. Now, black bears and golden eagles are 2 of our species, and so are great white sharks. Nobody's going to kill one. But the reason for that, I don't know how much you know about genetics, but when you're building these super high quality reference genomes, one of the things you want to be able to do is figure out where all the genes are and all the exon intron boundaries, as they say, the boundaries between the different bits of the genes. And the way you do that is by using RNA sequencing, because the RNA sequences inform you about those boundaries. And to do that well, you need lots of different tissues. So you need brain, and you need gonad, and you need muscle, and you need heart, and you need liver. And we do all that for people, but you need it. If it's a plant, if it's a tree, you can get root, you can get bud, you can get shoot, you can get flower, and that helps. So it sort of depends. But for everything else, for the other 149 individuals, I take a little tail tip. I take a little snip off the end of the tail. If it's a big animal, I can take a toe. I let it go, and that does it.
Okay, thank you. And I have a really quick related question, which was on the screen, and then I'll be done. How do you pick your reference genome? Does it have to be perfectly representative of the species? Like, what happens if you pick something that's sort of a weird outlier?
Well, I mean, in what sense will it be weird? I mean, the reason why you've got a reference genome. So a reference genome is this beautifully assembled genome, right? That ideally has chromosome level. You take all these little the bits of sequencing that you do, and you build the jigsaw puzzle out, and you glue them all together, and that tells you where every gene sits next to every other gene. It tells you where the promoters for those genes are. It tells you about the spacers between them. It tells you about everything about that genome. And, even if you're weird, I mean, that structure is a structure of the species. That genomic architecture is the architecture of the species. The one place where ideally you'd like to be choosy, and sometimes you can, sometimes you can't, is if you have a heterogametic sex. So if it's a mammal, we have XY in males and XX in females. You'd like it to be a male, because then you get both of the sex chromosomes. Whereas if it's, a female, you only get one. If it's a bird, it's the other way around. And reptiles and amphibians flip-flop. So, but other than that, I don't think you can be weird in that sense, right?
Great.
I mean, you might be,
Yeah.
anyhow, that's my answer.
Great, thanks.
Sure. Anybody else?
Jeremiah had a question about whether other states and countries are doing something similar.
Yeah. And Jeremiah, I don't know if you're there or not. I wish I could see your face, but it doesn't seem to.
I think.
Yeah, I'm here.
Okay. So the answer to that is yes and no. So there's a lot of work being done right now and lots of different projects being done that are constructing reference genomes for lots of species. So there's a very accomplished genomicist at UC Davis named Harris Lewin. And Harris is in charge of what he modestly calls the Earth Biogenome Project. And Harris's goal is to sequence a reference genome for every species of organism on Earth. He's got a ways to go, but anyhow, and there's a vertebrate genome project that does that, and there's the 10,000 bird genome project, and there's lots of different insect projects and stuff. All of them, it's just about getting a reference for as many species as possible. And that's fine. It tells you a lot about evolutionary history. It tells you about phylogenetic relationships. It tells you about how differentiated different species are. But I think the real sort of conservation value is in those 150 individuals. It's really not in the reference. I mean, if a pond turtle is different from a Sidewatch lizard, and that's, as an evolutionary biologist and herpetologist, I'm fascinated by that. As a conservation biologist, I sort of already knew that, and so it doesn't really help me a hell of a lot. Whereas those spatially distributed genomes across the landscape, they help a lot. They really provide that sort of definitive information. And to the best of my knowledge, nobody else is doing this. So it's, I don't know of any other projects that are like that. I'm having a call tomorrow with a colleague in Australia who's in charge of an Australian project. But I'm pretty sure they're just doing the reference genomes, at least to start. And it's a funny thing, because I think it's a fairly obvious thing to do. And in terms of the money, okay, we've got $12 million and for this project. And that's allowing us to do a lot. That's allowing us to push out a lot of money to a lot of people. That's allowing us to include a lot of species, more than we had originally thought. We can set up this core facility at UCLA so that people can just send their stuff to us and we can sequence it up for them. And 12 million is a lot. But, maybe this is just my jaded perspective from living in LA for a while. There's lots of houses that cost more than that. There's garages that cost that much down here. And so in the greater scheme of things, if you're a university person, that's kind of a modest upgrade to a building, to a building on your campus. And it's a potentially, I think, kind of policy changing data set for the most biodiverse state in the country. So I don't know, I think it's not a bad value. I mean, I think we can do it in part because we have the University of California we have this astonishing collection of people who are all interested in this kind of stuff and who have the capacity to do it. And maybe not all states have that. But I think, I mean, on the one hand, it's a sort of insurmountable sum of money for us, which is a sort of a sad statement on funding for some aspects of conservation. But in the greater scheme of things, it's not that much money.
It seems pretty cheap to me.
Yeah, I mean, it's not bad. And, we'll see if we can get this all to happen. But so far, it's all happening, and I mean, it's taken a lot of work. It's kind of all I've done for the last year and some change. And our executive committee and a lot of people who are putting in their time and energy, with no compensation, except that it's the science they do, and that's their job and that's their passion and that's what they love. And so anyhow, that's my answer.
Could I ask a quick follow-up on that? So like,
Sure.
would this data play nice? Say there was a study, a landscape genomic study on bobcats, like in other Western states.
Yep.
So you could probably directly compare, because it's whole genomes, the variation that you're getting in California with the variation of other Western states, or something similar.
Totally. So, and if they did whole genome, then they're 100% comparable. And that's where I was saying, if you each do reduced representation work, if you each do RADseq, it's really hard to compare those because, you get your 2% of the genome and I get my 3% of the genome, and they don't overlap much. And so it's very hard to align those and really do a comprehensive assessment. Now, if the Wyoming people do RADseq, which is a reduced representation, we can map their RAD data to our whole genome data. We can find those pieces. We can reduce ours in the same way as theirs and make them combinable. Okay, so that's one of the beauties of the whole genome data, is that whatever's out there, old data, scraps of data, you can always find it in that genome. And you can pull it out and you can make those side-by-side comparisons. But clearly the best is going to be if everybody's working at the whole genome level. And, I can't remember the genome size of a bobcat, but given that it's a mammal, it's probably about 2.5 gigabases. And, that means you can resequence a bobcat for $300. I mean, it's not that much, at 10x coverage, and that's not with the core sequencing facilities giving you any break on their sequencing. It's really getting pretty inexpensive.
We have a question too from Michael Goldman. Michael, you want to ask? You have a couple of questions, I think.
Yeah.
Yeah. Thanks for a great presentation and amazing work. What I wanted to know, like, how many individuals do you have to sample at one location to get a good estimate of variation? And
Excuse me.
it's difficult, to think about. You have to spread that out across, you have to spread 150 individuals out across as many locations as you're trying to sample. So first, how do you get those numbers? And second, I'm wondering how it is that you take an entire genome sequence and measure the difference between one and the other. Do you just pick out, say, 500 single nucleotide polymorphisms, or what is it?
Okay, so in answer to your first question, which is in my mind is the difference between landscape genetics or genomics and population genetics, right? So population genetics, we want to take a population, we want to sample a bunch of individuals, and we want to understand variation within that population. At the landscape level, what we want to do is understand, barriers to movement. We want to understand, those kinds of things that I've spent time talking about. So if you have a single individual from a site, the way I tend to think about it is something like this. For any given single nucleotide polymorphism, to use the term that you used, or any marker, an individual can either be a homozygote for one variant, a homozygote for the other. It can have two copies of one or the other, assuming it's diploid, or it can be a heterozygote. So its genetic variation at that site can be 0, 0.5, or 0, right? That's it. Not very informative, okay? Not very detailed. Look over 3 billion base pairs, and at the level of the variation that individual has, it's giving you a very fair representation of the overall genetic variation of 3 billion single, there may not all be single nucleotide polymorphisms, but let's say 40 million SNPs drawn at random from that population. That's a really good sample. They just all happen to be in the same individual, right? But you're getting a really good draw. So if you care about a gene, this is totally the wrong approach. If you want to know about genetic variation in some gene that really matters, this is the wrong approach. If you want to understand overall, are animals from here versus here versus here versus here more or less variable, and you're willing to just sort of pool it over the whole genome, it's fine. And you really don't buy much by having two, because again, you've got such a big draw already that it sort of doesn't matter. I mean, you gain a little more, but you don't gain much. I mean, the fall off is really extreme. So that's the answer to the first. And I'm sorry, remind me, Michael, what was the second?
Sorry, it was kind of, so I think maybe you answered it in answering the first, which is what you're measuring as you.
Oh, and you said, and also, right, how to sort of combine and what do you do? And no, you absolutely don't, within a species, you absolutely don't restrict it down to 500,000 or 500 or whatever markers. You take each one of those individuals, you remap them to the reference genome, you pull out every variable site, and then you use it to get an estimate of individual variation across every variable and invariant site in those organisms. And again, these tools have been percolating along on the human side, for a decade. Places like the Broad Institute in Boston, the GATK best practices, if you know those kinds of things, those are all there, and they're really easy to use. I mean, they're really not tough, and they're streamlined because why? Because the biomedical community wants to sequence every human on Earth. And so, they've got to figure out how to deal with 8 billion genomes. And they're beavering away at that. And we get to borrow that technology. And it's just like we get to store all of these genomic data for free on the NCBI, SRA archives, right? I mean, those are just things that come along, and they didn't build that for conservation. They built that for human genetics, but they're perfectly happy to share it. And so that's how you do it.
Thank you.
Yeah.
I had a question, Brad. Are you likely to find actually new cryptic species by sequencing so many individuals around the state?
I mean, and many of these projects, of course, I mean, who wants to do these kinds of projects? Who has, I've got 140,000, salamander and turtle and snake tissue samples in my freezers that I've collected personally over my career. Who does that kind of thing? It's oftentimes people who are evolutionary biologists who care about systematics. They do that kind of work. They're looking for cryptic species. They're looking to ask whether or not some subspecies is justifiable at the species level or not. And so a lot of our projects, I think the the folks who are doing, and I know, because I know a lot of these people, the folks who are doing it want to do it for exactly that reason. And I think, although that's not exactly landscape genetics, and so it's not exactly the kind of goal of the project, I think it is a very important goal. You know, if you buy into the notion that we only conserve what we recognize, and so if there's cryptic species and we don't recognize them, we don't protect them because we don't know, you know, we don't know that it's a thing to protect. And we protect entities, we protect species. And so to my mind, that's a perfectly justifiable outcome from the work. That's not necessarily the kind of outcome that when we take all the data through the CCGP pipeline that we'll be really looking for. But if the PIs are, that's great. You know, that's fine. And I expect there will be lots of them. I also expect some of them, certain taxa, people have a real proclivity to name things. You know, butterfly. I hope there's no butterfly enthusiasts in the crowd who are offended by that. But butterfly folks have oftentimes seem to be very enthusiastic about naming subspecies, some of which then get listed. And I would not be at all surprised if at least the genetic data pointed people towards saying some of those aren't any good. You know,
so you're going to be subtracting some species.
So you may well be subtracting some, and you know, if so, that's why we do science, right? That's okay. I mean, it may break your heart a little bit if it's one of your favorites, but I think that's just, you know, that's just the way it goes. So yes, I think that will. I mean, I know that will happen.
Yeah. Madeline Nash, you had a question, a couple of them, or maybe a comment about computer analysis of the data.
Mm-hmm.
Let's see. Oh, okay. Yes, I did. I think you mentioned before that there would be a lot of data coming in, and it does. Mm-hmm. So I was just wondering. It seems like it calls for some major computer power. Mm-hmm. I was wondering how that effort is going, whether it's underway yet, and how it's being approached.
No, it's a very important question, Madeline, and a very relevant one. And I think that so in terms of dealing with the data, you know, that a lot of it has to do with getting data streams off the sequencers and how long you retain the raw data versus, you know, turn it into more curated data and then throw out the raw data and keep the more curated data, and how quickly you get it onto the big databases like the NCBI database that the federal government supports for storing it. And that's okay. I think that's going to move along pretty well. What I think the real challenge is going to be, so, you know, I think the computational goal on this is, in my mind, is to be able to do a lot of analyses and then store it in a well-organized, curated way. And it's not just the genomics data, it's also all the landscape layers, all the GIS layers and stuff. So that a person, you know, my vision is that you can have a base map of California and you can draw a square around an area of any size. And on the fly, you can say, show me a landscape genomic map of that region. And then you can say, okay, show it to me just for vertebrates, just for trees, just for insects, because that's what I care about. And you can sort of do those calculations on the fly. And that, I think, both in terms of storing it in a way where it's accessible to anybody over the web, and being able to have those sort of scripts written so that it can do those computations very, very quickly. I think that's going to be the computational problem. And having it be long-term, you know, it's fine for us to say genomic data is valuable long-term, but it costs money to store it. And I'm looking into a couple of different options for that. We have a partnership that I mentioned with the California Department of Food and Agriculture, and they store and curate enormous databases on pesticide use and other kinds of things in the state. I'm hoping maybe we can partner with them for a sort of long-term storage and data repository kind of thing. So, but to me, that's the really big computational thing. If we had to keep it all ourselves, if we didn't have the SRA, then we'd be in deep trouble. But we do, and so it's a matter of just having a flow so that we don't have too much of the data sitting on our shelves all at once. We can sort of move it through, move it out, move it through, move it out, that kind of thing.
Thank you.
Yeah.
I don't think there are any more questions.
Okay.
So if somebody has one, you can pop up now and ask it. But
can I just
think
ask a question about the RNA-seq?
Sure.
You mentioned that you were doing RNA-seq on multiple tissues.
Mhm.
I was just wondering how comprehensive that was for these reference genomes. Will there be tissue-specific RNA-seq available?
There will. So it depends on what the PIs give us, right? So the RNAs are going to be extracted and sequenced and kept track of at whatever level the PI gives it. So if I give a liver and a heart and a brain of a snake, I'll get those reads back. In terms of using it to annotate the genome, I mean NCBI actually does that annotation automatically, and I don't think they do tissue-specific annotation. I think they're just using it to get transcripts to kind of look at where the exons are. And so in that sense, since that's our main goal in this project, what we're really trying to emphasize is, I mean, we're keeping the tissues separate, not because we care about tissue-specific transcripts for every species, but because if there are rare transcripts in one tissue, by keeping it separate, you've got a better chance of actually sequencing the damn thing. And so that's why but it will be there for PIs.
Okay, that makes sense. Thank you.
Okay,
Yeah, really
sure.
cool project.
Thanks.
Lisa Krieger has another question about.
Yeah, and I'm
rules.
sorry, this is so far afield from what you do, but you're like the first biologist I've talked to all week. So I'm just curious. The wolf that left the Mount Hood area and now it's in San Luis Obispo County.
do,
Like
yeah.
how crazy is that?
Mhm.
Do they hybridize with coyotes? Like what?
They do. They certainly do in the eastern wolf up in Canada, up in southeastern Canada. There's that endemic. I can't remember what it's called now, but it's around Algonquin National Park is a separate taxon of wolf, and I know they hybridize a lot with coyotes. And I think they can hybridize anywhere.
They'll get established in that neck of the woods and just hunker down.
Yeah, I mean, I mean, my guess is that if he's wandering around looking for a mate and there are no wolves around, I wouldn't be surprised if as long as he didn't eat the coyote first, I wouldn't be surprised if they mated. It's I mean, that's if you're a biologist, that happens all the time with it's LE effects and stuff. So that happens all the time. I mean, hopefully, in my mind at least, hopefully that won't happen. And what will happen instead is a few more of them will make it down, or come over the crest of the Sierras. And I mean, from the Sierra side, it's going to be tough to get into San Luis Obispo County. But if one did it, and if we can just keep our hunting enthusiasts at bay for a while, they'll get in. If we give them the opportunity, there's that wolf that went from, Detroit to Massachusetts in a month or something. I mean, they can really move when they want to.
And what's the likelihood that he could turn around and find his way back to his?
I don't know. I don't know.
Okay.
I know who would know, my colleague Bob Wayne here at UCLA, who's studied canids and wolves in particular his entire career. My guess is that he probably could. And, but I just base that on the fact that, canids in general are just so amazing they're good at migrating, they're good at homing, they're good, they're good at that stuff. So I bet he could. I can't imagine why he'd want to, but maybe. Anyhow.
I can't remember if you mentioned that you're collecting poop for some of these hard to collect, hard to find animals.
Me?
Or your colleagues, is that a source of DNA for you?
It's a source of DNA for doing things like environmental DNA or DNA barcoding. So if you just want to get, I mean, the way poop samples or hair samples or a lot, environmental DNA samples usually work is that generally, at least for most, for metazoans, for animals, the mitochondrial DNA is present at, whatever it is, 1,000 times more copies per cell than nuclear DNA. And so usually you go for a mitochondrial gene and you can use that to sort of barcode something. You can use that to identify it. And within poop, you can do metagenomics and you can, sort of randomly sequence up what's in there with some of these kind of barcode genes and see what that coyote's been eating, how much of it's plant, how much of it's animal, that kind of thing. But you can't do genomics on that stuff. It's too degraded, it's too fragmentary. It's just, you have to have something that's reasonable to work with. It's all what's called short read data. So it's all 150 base pair fragments for the resequencing. So it's not like the DNA has to be in big, long chunks, but it has to be, it still has to be reasonably good DNA in order to get reasonably good coverage, to get a reasonably good genome.
Okay.
Yeah.
I think our last question will be from
Okay.
Michael. He had a final question, he said.
Yes, and this might be a strange one, but are you getting any epigenetic data along with your sequence reads? And you said this is the end of the line for new technologies. What if we start to find some epigenetic variation that has significance at the conservation genetics level or at the species?
For sure. And I mean, when I said it's the end of the line, it's the end of the line in terms of sort of understanding the genome. Now, also, Michael, since you seem to know some stuff about genetics and genomics, 10x coverage is sort of marginally good coverage for calling heterozygotes. Many people want 20 or 30x coverage to be able to do that more accurately and more completely. And so is it the end of the line? Well, no, not really. But the nice thing about that is that you can just sequence some more of it and get higher coverage. So, if you still have the sample, so that's nice. It's very combinable. On the epigenetic side, I fully admit here, you're kind of pushing my boundaries. And I think you get methylation data out of it, and that sort of is tied to epigenetics, but it's not exactly the same thing. So I think you get some, or you can. You also don't get expression data, and as many people have said, well, maybe it's not about the sort of baseline sequence. It's about expression. How much are you over or under expressing different genes that are important for resistance to this, that, or the other thing. So there is always more, but at least in terms of the main sort of gold standard that we've been going after since the days of Alzheimer's, it's always been to try to understand the genome, the sort of, core sequence that is your chromosomes. And for that, at least, I think we're kind of getting close to an endpoint. Fully admit there's other things that, like we just talked about.
Well, thank you, everybody. Thank you, Brad, very much for a very interesting talk. And we hope to hear more about the final results. When will those be in? And about
well, I mean, I think, you know, I think in 6 months, we're going to start having really good data rolling in, and a lot of it. And I think in a year and a half to 2 years, we'll have it all. That's my goal.
And let's hope the state will act on that information.
And I think they will. I think if we make it, accessible, they will. I think if we make, and that doesn't mean, you know, dumb it down or anything. That just means make it that, you know, a reasonable person like all of us with some, you know, reasonable level of interest can actually use it to make decisions. And I think if we can, you know, I don't think, at least I have lots of colleagues in the Bureau of Land Management, for example, you know, and some BLM land is open to off-road vehicles and some BLM land is wilderness and you can't take a bicycle into it. And I think if we could guide the BLM on which were the most important pieces of their millions and tens of millions of acres that they own in the state of California, and give them some guidance, I think they'd shift it around. You know, I think they'd move wilderness status over to other areas. So I think they would. We'll find out.
Fingers are crossed.
Okay. All right. Well, thanks a lot, you guys. It was great questions and great fun. And I thank you very, very much.